Office is right next to this building, right over there. Yeah. That picture was taken by Steve Pinker. Out of Jack Loomis was flying the plane, and Steve was taking pictures out of the plane window. Okay, I wanted to talk today about work that we've done on perceptual categorization and everything from the initial learning of categories to how they become automatic. And I think it's going to tie in with stuff that we've been hearing because the role of language, I think, oh, I'm out of the picture. I have to be in the picture too? Sorry. Okay, sorry. The role of language plays a big role in the way we think about these problems. So I'm going to start off the first half. I'm going to talk about the initial learning. We focus on two different kinds of tasks because basically we have lots of evidence now that there are multiple category learning systems. One of those systems depends very critically on language, we think, and the other one doesn't. And then I'm going to talk about some more recent work on how we think some of these very difficult kinds of judgments become automatic. So we've had a long history of this kind of research, and we've used many different kinds of stimuli, but this is the kind of stimulus we're using now. These are derived from vision science Gabor patterns. Where the two ways that these patterns vary across trials is in the width of these bars and in their orientation. And there's a couple reasons. There's actually a big, long story on this I'm not going to get into, but there's the two reasons I want to mention why we use these very artificial-looking images. One is we're very interested in initial learning, and so we don't want to use stimuli that people have prior history learning, because that could interfere with the structures that we're examining. The other has to do with language. And it's very important for us in our analysis, and this will become clear later, that we can identify from response patterns that we get from our subjects whether they might be using a strategy that they could verbalize versus one where they can't. And for example, with things like rectangles, which we used briefly, there are many different kinds of strategies that people can verbalize. They can talk about the height of the rectangles, the width of the rectangles, the size of the rectangles, the shape of the rectangles. And so you can get very different kinds of behavioral data from subjects, and it's very difficult to tell whether they actually might be using a strategy that they can verbalize. With these stimuli, we've run hundreds of subjects with these. There's really only two things that people can talk about, or at least they seem to talk about. One is the width of the bars, and the other is the orientation of the bars. And everything else that they might talk about, we don't vary. And so that really pretty much limits the kinds of verbalizable strategies they can use, which helps us when we analyze the data to know whether they're using verbal rules or rules that they can't verbalize. So they come in and we tell them, you know, the prototypical experiment, there's two categories of these disks, and they're going to vary on these two ways. And here's two buttons, A and B. The first disk, the first image they see might look like that. They have no idea, of course. They press a button. We give them, typically, we'd give them feedback about whether they're correct or incorrect. And then they just keep proceeding. Get another example, and they do this over and over and over. Anywhere from sort of a minimum of 600 trials to 15,000 trials, depending upon what the experiment we're doing. You have to pay people if you're doing that many. We create different experiences or different category structures for subjects depending on how we divide these disks into the two categories. And here's an example, what we call rule-based category task. And it's rule-based because there's a simple rule that's easily verbalized, which divides the disks into the two categories. It's just, in this case, the thick bars are in B and thin bars are in A. And as you can imagine, people quickly learn these categories. They can learn here I've shown, I don't know how many, 8 or 10 in each category, but in the way we actually do these experiments, there's usually a unique stimulus on every trial. So we usually sample from this whole space, a unique point every time. So they never see anything twice, so that slows them down a little bit. But even so, most people can learn these categories in about 80 trials. They're essentially perfect. At the end of that, if you ask them what they're doing, they very accurately tell you, I was just assigning thick bars to B and thin bars to A. So these kinds of tasks have been studied in psychology for, 60, 70 years. I'll get into this more later, but for example, this is another thing I want to show is that it's not simply the fact that it's a one-dimensional problem, but it's that it's easily verbalized. That we think is the important thing. So here's an example. We would also call this rule-based. This involves both dimensions, but it's still pretty easy to verbalize the rule. Here it's just disks that have shallow orientations and thin bars are in A. Everything else is in B. So it's a conjunction. Learning of these kinds of categories requires no distraction. So if you have a dual task that people have to perform simultaneously, they're actually impaired in learning these kinds of categories. I'll show you some data on this in a minute, but processing the feedback signal requires attention and effort. But the nature and timing of the feedback is very flexible, not critical here. Okay, now, and I think this is going to map on to sort of color naming, where we have these names that go with the categories that the culture uses, and that's exactly what's going on here. But not all categories that we learn have names or are associated with verbal labels. And so we can take the same exact stimuli and divide them differently. And in English, at least, or in no other language I've ever come across, is there a description of this category boundary. So there's no conjunction or anything like that that's going to work. And remember, even if you could cook up some complicated thing that would work for these 12, remember, they're really seeing hundreds and hundreds. So there isn't any way to describe, using words, what's being learned here. People can reliably learn these categories. For sort of healthy young adults, it takes about 800 trials instead of 80 trials. So it's about 10 times as much training. But at the end of that time, people are very reliably and accurately, yeah. Question?
So there's not more spatial frequency in A than there is in B?
Well, you have some very thin, high spatial frequency here. There's also some very low.
I mean, couldn't you do an overall? Because it looks to me like, the average spatial frequency of A is greater than B.
Yeah, that's going to get you above chance.
Okay.
So, for example, that would just be a vertical bound here. And the vertical bound is going to miss these and it's going to miss these. But you can get, you could probably, it depends on how we set up the categories and how close they are and how much. Sometimes we have overlap, but you might get 75% correct with that, whereas you could get 100% if this bound would give you 100%. You might get 75 or 80% with a verbalizable rule. So verbalizable rule will get you well above chance, but it's not going to get you perfect. If you tried to verbalize the rule, it would be if the orientation is greater than the bar width, it's in A. Otherwise, it's in B. Well, right. That's not anything any of our subjects have ever told us. That's the mathematical formulation.
Orientation is an angle, and the bar width is any number.
Yeah, that's right. That's not a verbal rule. That's right. Yeah, that's right. That isn't anything any subject has ever described to us. Instead, if you ask people what they're doing at the end of this, you get a variety of things. Some of them don't make any sense. But often you'll get things, I just went with my gut reaction. Or we have subjects say, well, I just started singing to myself and pressing buttons, and suddenly I was getting things right. So it's a very, very different qualitative experience from the subject's perspective.
But I still get it. If I was a machine doing this test, what rule would I apply to the stimulus to know which side of the part it is?
Well, these are ratio scales, and so it's just y minus x, but you have some scaling problem, and that would work. If you give these data to any cluster analysis routine in SPSS, you have to obviously input them in some numerical value, but SPSS is going to have no trouble separating these stimuli.
So it seems like the critical issue is not so much verbalizability, but rather whether the things are verbalized by subjects. Is that right?
Well, you know, I'm not a linguist, and so when I first started going around in conferences and using verbalizability, I would get hammered by people who knew a lot more about that than I did, and so I sort of have backed off on that. That's the criterion we use in the lab, which is sort of an operational criterion that we use ourselves to decide which of these two, and of course you're going to anticipate what's happening. Different brain systems are going to be learning these two different kinds of categories, and that's the operational criterion we use. I'm going to show you some non-human data on monkeys and pigeons in here, and maybe that'll speak more to this issue. But I don't think it's exactly verbalizability. I would identify it more in terms of brain regions. So this is, the other system is going to be more of a prefrontal cortical region or task. Yeah.
Aren't there some stimuli that are close to that boundary in which they're making lots of mistakes?
Yeah.
And never really learning?
Yeah, well, you can, that's right, because there's perceptual limits. But we can, again, we've run so many of these experiments that we have all different variations. So and sometimes here I show a gap between the two. In others, they actually overlap so that optimal performance might be only 90% correct or even 80% correct. And but that doesn't really seem to change any of these qualitative properties. It changes sort of how quickly they learn. It changes things like that, how accurate they end up becoming, but it doesn't really change, I think, which brain systems are being used to learn these categories. Now, these kinds of categories have not been studied nearly as much in psychology as the rule-based kinds of categories. But our argument is many real-world categorization tasks involve, I think, both kinds of learning, but certainly this kind of learning. And here's an example, sort of a real-world example that might involve this kind of a very similar kind of task. This is a mammogram, and the radiologist's job is to decide whether there's a tumor in the mammogram. And I'm not a radiologist, but I'm told that in fact there are two strong possibilities of tumors here. And you can now an expert radiologist can very accurately identify these two spots, but you could imagine the difficulty in writing a verbal description which would allow non-experts to pick those two spots out and no other spots. So our argument would be that this kind of a task, clearly there's explicit learning here too. That's what you learn in medical school, but you don't become an expert radiologist in medical school. You have to go on residency and you get hands-on trial-by-trial training. And so that's the component, I think, that involves this more, what we're calling information integration. As I said, people can learn these categories. There's some very big qualitative differences, and I don't have time to go into a lot of these today. But for one thing is that this kind of learning is not really very much affected at all by a dual task. So you can have a simultaneous dual task that interferes with learning a simple one-dimensional rule, but it doesn't cause any significant interference with this far more difficult problem. Another interesting thing about this kind of learning is it's more tied to motor responding. So we did an experiment where people learned these categories, either learned this kind or the rule-based kind. And one group actually, during training, they had their fingers like this and they uncrossed their hands. Another group didn't change their hand positions, but the button positions reversed. None of those manipulations had any effect on their ability in the rule-based condition. They were just as accurate after the change. But in this condition, it turns out that uncrossing their hands had no effect. There's no interference caused by that. But switching the buttons caused a massive interference. So the argument is that they're not learning a motor act because the motor act has changed when they uncross their hands, but it's a higher level motor goal, which is press the button on the left versus press the button on the right. And you uncross your hands, it's still the same motor goal, but if you change the buttons, the motor goal has changed, and that causes interference. Okay, so These are the two kinds of category structures, and again, if you were to input these in SPSS or in some stat package, the cluster analysis routine, I don't think would care which one it was working on. But certainly reviewers care, and reviewers are very strongly convinced because, again, humans, normal humans takes about 10 times as many trials to learn these categories as these, and reviewers are strongly convinced that there must be something inherently simpler about this kind of category structure than this kind of category structure. So the question is, are humans 10 times slower here because of something inherent to the category structure out in the world, or is it because of the brain systems that they're using to learn these? And you know, I'm going to argue the latter. But the question then is there some inherent difficulty difference here, or is it just different learning systems? And you know, the argument here is, well, there's only one dimension, and here there's two, and so maybe that means that this is an inherently simpler task. I'm going to argue this is simpler for us because we have a large prefrontal cortex that we can use here that doesn't really help us out so much over here. And so one way to address that question would be to look at animals without a prefrontal cortex. And so here's Gary Larson's take on animals without a prefrontal cortex. Well, time for our weekly brainstem storming session. Right, so we're told that we don't have dinosaurs, but we have birds, and supposedly birds evolved from dinosaurs. So we ran pigeons on exactly the categories that I showed you. The discs, diagonal category, simple one-dimensional category, exactly on these two category structures. The birds had two keys. One was red for category A, peck red for category A, peck blue for category B. They get, of course, food reward. They get about 70 trials a day, and then they're full of seed, and they don't want to work anymore. So it takes many, many days to do this. And here are the learning curves. This is run in Bob Cook's lab. Here are the learning curves for the two conditions. Pigeons don't care if there's a one-dimensional rule or if it's the diagonal rule. They learn at exactly the same rate in both kinds of category structures. So we would argue that on the basis of that, that would sort of verify there's no inherent difference in the external category structures. The difference, this huge difference in learning rates we see with people is because of the different brain systems that are being used in the two kinds of conditions. Now, we also have more recently run monkeys, and this is in Michael Barron's lab. These are 6 monkeys that ran in the information integration task and in the rule-based task. Again, exactly the same stimuli and categories that the pigeons ran in. Each animal here did both conditions, and half of them did one first and the other one second. And monkeys, these are all on computers, and monkeys apparently love to work on computers. You get 2,000 trials in a day from a monkey. You can't get that many from a UCSB undergraduate, but monkeys are happy to work away. But they're also very social, and if they're at work and they know there's some important social activity in their colony, they don't work very well, and they're kind of distracted. And so apparently, I think that's probably what was going on here with Luke. Because this is, Luke didn't really learn the rule-based. But so there's variability here. But if you look, there's 4 of these monkeys learn these rule-based categories very, very quickly. And there's some evidence of very quick learning here. But generally, the learning is much slower with the information integration categories.
A couple of these seem to know the rule to begin with.
This is because Murph, for example, Murph over here had some learning of the information integration rule and was initially applying that. And that's going to get above chance accuracy, right? And so instead of starting with a blank slate, there's already a certain, maybe 60, 70% correct, and then bootstrapped up from there. So the important thing is sort of the learning. Well, also, I mean, this is very, very quick. But again, you can get, if you run this on humans, you're going to get some humans that are perfect within 15 trials. All right, so the monkeys sort of look intermediate between pigeons and humans. All right, so So now the theory of learning that we actually proposed a long time ago, more than 10 years ago, but the idea is that in these two tasks, there are these two systems. One is this explicit logical reasoning system. This is a system that, again, in psychology is very, very old, goes back really to Aristotle, and Spence worked on this and lots of other people in the 50s. But the idea is that people generate explicit hypotheses about category membership. Well, I think it's thick bars versus thin bars. They test those hypotheses. Feedback either confirms or rejects. The problem is that system is not going to work very well in the information integration category problem because there isn't a hypothesis that it can generate that's going to work. And then there's this other system, this which we think is procedural or habit learning system, which learns more slowly, and it can learn virtually any similarity-based rule. And so I think this is maybe how the pigeons were, something similar to the way the pigeons were learning these categories. Yeah?
I'm wondering if there was, whether some, in these slow learning situations, if there was some information that was actually correlated with the right category, but that the subject wasn't aware of. In other words, maybe lights flicker or something in the surround that actually, whether or not their behavior would reflect that correlation without their having any sense at all that was entering into it.
Yeah, whether it's implicit learning that's going on here?
It's even beyond that. Like, they're looking at these little boards, let's say, but suppose you fool them. You make the right answer dependent on, whether or not there's a little sort of like plum in the radiator.
Yeah.
So, it might be that in fact, given that if you say that they're sort of trancing out when they're doing this, well, maybe they're being controlled by all kinds of cues that they are not even aware are relative to that. I mean, have you ever looked at that at all?
We actually have a study now going where we've embedded a sequence into the response buttons. 10 buttons in a row have they're random, but then that random sequence repeats. And the first subject we ran comes out, oh, you had 10 buttons in a row, and here's a sequence. But most subjects are not aware of this, and actually, but their accuracy goes up, so they have implicit learning of this sequence. And then we randomize, take the sequence away, and we want to see, did that actually help their category learning? But it just drops right back down to the same place that other subjects. But I think that's right. I think this system is operating on many different cues that are coming in, and what's happening is it's sort of figuring out which cues are relevant and predictable, but there's not a lot of explicit awareness about this on the part of the subject. Now, the reason I don't use the word implicit, because for those of you who know this literature, sort of hardcore implicit learning theorists have two very strict criteria that need to be met before they'll acknowledge that it's implicit learning. The first is that people cannot describe what they've learned. We clearly meet that condition because none of our subjects can tell us what they've learned. But the second condition is often that, in fact, people don't even know they've learned anything. Now, that condition is impossible to meet in these experiments because they're getting feedback on every trial, and the feedback's telling them they're getting things correct. So they can make the inference that I must be learning something because the feedback is telling me that. So I don't really, use the word implicit. I think it's procedural learning is the word that I would prefer, but it is very much in that same flavor. Okay. Now, the neurobiology that underlies a lot of this depends on much of it depends on a system, a structure in the basal ganglia called the striatum. So the basal ganglia are a large collection of subcortical nuclei. The striatum includes the caudate nucleus, is the major input structure, and then also the putamen, which I think has very similar kinds of properties. Here's a human dissection that shows the caudate nucleus. This is the front of the brain, prefrontal cortex. Here's the visual cortex. And when people talk about the caudate, they often talk about the head and the tail. And this is actually the fiber bundles projecting from cortex into the caudate. And you can see that, in fact, all of cortex, in fact, in humans, all of cortex except for V1, primary visual cortex, projects directly into the striatum. So the striatum is getting direct input from all of cortex. So it's in a very unique position to mediate some of this kind of learning. There's a whole bunch of evidence that the basal ganglia are critical in these tasks, and in fact, we just had a whole chapter on the role of basal ganglia in category learning. I don't have time to go through most of it. I just want to show you one study which sort of is evidence that the striatum is important. This is a study that Romo, Merchant, and their colleagues reported. And a monkey's got its finger out. There's a stylus on the finger, and the stylus gets pulled across the finger at one of 10 different speeds. And if one of the 5 low speeds occurs, the monkey is supposed to push one button. One of the 5 high speeds occurs, it pushes the other button. And as soon as the animals had learned this or acquired these categories, these guys did single unit recordings in the striatum. This is in the putamen. The putamen, because the putamen is the major input, is somatosensory cortex, which is presumably mediating the perceptual representation of these stimuli. And they found lots of different kinds of cells, but they found a category or a class of cells that fired to category membership. So here's a cell that, here's the 10 different speeds, and each row is a different trial, and each dot represents a spike from the cell. And you can see that this cell is firing to virtually all of the low speed category members, but not to the high speed. And here's a cell that has the opposite pattern. It's firing to the high speed members, but not the low speed.
And these cells did not behave this way prior to training?
Well, they didn't do the recording prior to training, but they did a bunch of tests to make sure that, for example, they weren't just responding to the movement. And when the animals move in the absence of the task, these cells don't show this pattern. When they're passively presented with the same stimulus, they don't show this response. Okay, so just a little more detail about the system. So the logical reasoning system, we think, uses working memory and executive tension. The idea is you have to hold these rules in your working memory. And while you're testing them, you allocate executive attention to different parts of the stimulus. And we proposed a network that we think mediates performance in these tasks. And I don't have the time to go into a lot of detail here, but basically the idea, this is much of this is a working memory model. It's very similar to other working memory models in the literature. But so working memory is thought to be mediated by reverberating loops. And in this model, there are 2 reverberating loops. One to the posterior cortical area, which sort of mediates your long-term memory of what this rule means. And so this might be, for example, Paul had the color naming areas in parietal and temporal cortex. That might be this unit. And then when you're holding a rule that would use those same color names in memory and you're trying to apply it, there's would be reverberating activity into prefrontal cortex working memory units. And that activity is also mediated by another reverberating loop in the medial dorsal nucleus of the thalamus. And there's been a lot of working memory research that basically supports a model of this general type. So you're holding then this rule in working memory. Feedback then tells you that you're wrong. So then you have to, okay, this must not be the right rule. I have to try a different rule. So you have to select a new rule, and here that would be mediated by a circuit anterior cingulate and prefrontal cortex. And you have to switch attention from the old rule to the new rule, which is mediated here by prefrontal activation of the caudate nucleus, which essentially breaks this loop and allows you to switch attention. Transferring the information long-term memory involves projections into the hippocampus, and a lot of these processes are facilitated by dopamine. The procedural learning system is very different. And so here, the key structure is the striatum. And there's 2 key properties that we think are important for why the system is so critical in these information integration tests. One is not only you get the input from cortex, but there's massive convergence in this input. In fact, it's about 15,000, between 10 to 20,000 to 1 is the convergence. So approximately 15,000 cells in sensory association cortex will project onto each cell in the striatum. And the idea is that, remember, in our experiments, every trial is unique. But what's happening is you're training up the striatal cells so that all these cells from sensory cortex that are projecting in there, eventually, even if it's a completely unique stimulus, it's projecting into a unit that's already been trained up. So that's one key feature. The other key feature is that there's dopamine release into this area from the substantia nigra. And there's a lot of people who have argued that dopamine serves as a reward-mediated training signal in the basal ganglia. And the idea is that following correct feedback, dopamine gets elevated, and it's going to strengthen recently active synapses. Following error trials, dopamine will be depressed below baseline levels, and that will weaken recently active synapses. And through that process, you can learn these kinds of arbitrary stimulus-response associations. Now, if you, I'm going to focus in now on this one synapse. So this is synapse again in the striatum. And here it is. So here's the terminal end of the axon that's coming from sensory association cortex. This is the dendrite of the medium spiny cell that's intrinsic to the striatum. And this is the dopamine cell coming from substantia nigra. And again, there's a lot of evidence that the dopamine actually gets released into the same synapse and affects the same synapse. But if you think about this story, there's a big timing problem. And the timing problem is that the subject sees the disc, which presumably causes the visual cortical cell to fire, which then causes this cell to fire, and then there's a train that a sequence that gets carried out causes the subject to press say the A button, then they get feedback that was the correct response. That causes dopamine release because we know how much our subjects want to please us and get everything correct. And then the dopamine gets released. But the dopamine is now being released sometime after this cell was active. So how does the dopamine know which synapse was the one that was responsible for the reward? And there's a very clever solution to this problem, and that is that these cells are called medium spiny cells, and they have all these little spines. And the spines are critical here because what happens is after this cell fires, there are natural processes that reset the membrane potential. But the spine is actually physically separated from the main dendritic compartments, and so those processes take longer to reset the membrane potential in the spine. So it's like if you dump dye in a river, the main channel gets colored very quickly, but the little tributaries, it takes longer for the dye to diffuse in there. And the same thing happens here. And in fact, there's neuroscience evidence saying that it takes about 2.5 seconds to reset the membrane potential in the spines. So there's a very, very strong prediction right here, which is if the feedback signal is delayed by 2.5 seconds after the response, this charge will have been reset, and the dopamine gets released. It has no idea which synapses were responsible, and there shouldn't be any learning. So we did that experiment, and this is the structure. That we use these same Gabors. So in the immediate condition, the subject would see a stimulus, and it would be up there until they hit either the A button or the B button. Then we'd mask it with another Gabor for a half second, then they'd get told whether they're correct or incorrect, and there was a 5-second delay between trials. In the delay condition, they see the stimulus. It's up there until they hit A or B. Then they're masked, but now the mask is on for 5 seconds. Then they're told they're correct or incorrect, and then there's a half-second delay, and they go to the next trial. And here are the results. For the rule-based categories, no effect of the delay. For the information integration categories, there's a big drop in accuracy. In fact, they're just barely above chance. We replicated that experiment. That was a 5-second delay. It also exactly the same results with a 2.5-second delay and a 10-second delay. But this is actually pretty amazing because this is saying that the rule-based category learning, there's no impairment. Even if you have to wait 10 seconds to be told if you're correct or incorrect on every trial, you learn just as quickly as if you get immediate feedback. But of course, the information integration is very, very different. We also varied the level of difficulty of the rule-based tasks, that didn't matter. And then later, Todd Maddox ran another version of this where instead of 2 categories, there's now 4. These are rule-based because, again, these are just conjunctions. So these are thin bars that are shallow. These are thick these are thick bars that are shallow. These are thin bars that are steep. And so there's simple linguistic labels with those categories. You just rotate everything. Now you can't verbalize this. But now the rule-based, both of these depend on 2 dimensions. And he gets exactly the same results. I don't remember what the delay here. I think it was around 3-second delay. There's no effect on rule-based learning whether there's a delay or not, but the information integration is significantly impaired if you have to wait for the feedback. Now, so that's a big advantage to the rule-based system because you can get the feedback much later. We also did another study. You can give the feedback before you even show the stimulus, which is what, of course, teachers do in class, or what we do as professors. Okay, here's an example of this condition, and then we show them the stimulus. Rule-based learning is fine there. Information integration learning is terrible. Jack?
I was wondering, why do you think there's any limit?
Well, remember that because the rule-based system can get you above chance in these tasks. And there can be rule-based learning with delayed feedback. In fact, if you do the modeling, that's what's going on, is these people are, their behavior is described by rule-based learning. Okay, so there's a big advantage then with respect to how feedback is treated to this explicit system. But the other system has advantages too. And so in particular, the explicit system has to think about what the feedback means. So if I'm using a rule like thick bars versus thin bars, feedback tells me I'm incorrect. I have to go through this very effortful process. Okay, it must not be the thickness of the bars. What else could it be? Maybe it's the orientation. Maybe steep bars are A and shallow bars are B. That takes time, attention, and effort. Whereas the implicit system, you're just singing a song to yourself, pushing buttons. Dopamine squirts out, automatically strengthens the right things. You don't have to think about what it means. And, that's a big advantage there. So we tested that in this experiment. Again, Gabors, they get a Gabor, then there's they get the feedback. And again, this is all response-terminated displays. They get feedback. And then there's a half-second delay, and then we switch them over to a completely different task, which is a Sternberg memory scanning task. Nothing to do with categorization. They just get a sequence of numbers. They have to memorize that for a second, and then they get another number, 7. Is that in this display, yes or no? In this case, no, so they hit the no button. Then they get 2.5 seconds, and they get another categorization stimulus. Same exact thing here. The only difference is in the delay. So here, there's a 2.5 second delay after the feedback and before the memory scanning. And there's yeah, this is wrong. I think this should be, because the total time is the same, this should be a half second, not 2 seconds. So the question is, does it matter if you have a half second to think about what the feedback means or 2.5 seconds to think about what the feedback means? Exactly the same categories we've been talking about. In the information integration condition, this is a control where there is no second task. Just do categorization whenever you talk. It doesn't make any difference if you have a half second or 2.5 seconds. You got to get that. You get the feedback immediately, and you learn fine. It doesn't matter if you have to transition into a different task right away. But in the rule-based condition, with a short delay, a half second, you're significantly worse in your ability to learn these categories. So both systems have advantages. They just have different kinds of advantages. There's been a whole bunch of studies that support this general model, and I'm not going to go into any more detail here because I want to talk about automaticity, how these kinds of behaviors become automatic. But it comes from lots of different kinds of studies, single unit recording study, and I showed you some of that already, animal lesion experiments, neuropsychological patient studies. Some of these were explicitly run to test the theory, some weren't, but they all generally reported results consistent with this theory. Neuroimaging data and traditional cognitive behavioral experiments. So what I want to do now, though, is talk about how these kinds of behaviors can become automatic. And I think that's certainly the case with color naming because people have had a lifetime experience with that task. So here's our family dog, Coltrane. And the point here is simply that you see a dog instantly and not a bear. But if you think about the features that this guy has, he actually shares lots of features with a bear. If you tried to, say, do an AI program or model to discriminate, it wouldn't be simple. But we can do this immediately. The question is, how does this become so automatic? And if you look at the neuroscience literature on automaticity, it goes back at least to Charles Sherrington, who's the guy who coined the term synapse. And so he says, as I write, my mind is not preoccupied with how my fingers form the letters. My attention is fixed simply on the thought the words express. But there was a time when the formation of the letters, as each one was written, would have occupied my whole attention. Now, Sherrington worked on reflexes. And so he actually believed that automatic behaviors became reflexive. And that more complicated behaviors where you're just stringing together a sequence of reflexes. So he actually thought that some of this was mediated in the spinal cord. That didn't last long, but his influence was dramatic. And in fact, the dominant theory throughout the 20th century is illustrated in two quotes here, one from Carl Lashley's famous paper in Search of the Engram. It's been widely held that although memory traces are first formed in the cerebral cortex, they're finally reduced or transferred by long practice to subcortical levels. And here's a similar quote from Fuster, much more recent, showing there's still a dominant theory. Now, I've been arguing that the initial learning is subcortical in the basal ganglia. So, in fact, I'm going to argue exactly the opposite of these theories, that the initial learning is subcortical, and with practice, it moves to cortex. So it's exactly the opposite of the theory that expressed here. One question is, well, why do we need a different theory? What's wrong with the theory that I've already shown you? And there's several reasons to think that the theory that I've shown is inadequate. One is that if let's take patients with basal ganglia dysfunction, so Parkinson's disease or Huntington's disease, as the theory predicts, they are massively impaired in learning new categories. But they don't lose old familiar categories. So Parkinson's patients don't forget that this is a table and all the other millions of categories that they've learned throughout their whole lives. They only have trouble in learning new categories. You can contrast that with another group, and there's a group who have category-specific agnosias. This is usually caused by some lesion in cortex. The most famous example is prosopagnosia, and these are people who have a difficulty identifying and categorizing faces. Now, I've got a question mark here because I don't know if anybody's done this study, but from talking with prosopagnosia researchers, my impression is that these patients don't have a general category learning deficit. They only have trouble with a specific class of stimuli that, for example, with faces or some of them man-made objects or tools or things like that. So you have this potential double dissociation as a problem with the theory that I just described. So the question is, how could a behavior like this ever become automatic? And if you think about it, it's a visual stimulus, so we need activation, initial activation in visual cortex somewhere. A motor response is produced. There eventually has to be activation in a motor area. And I argued that the way the signal gets from visual cortex to motor cortex is through the basal ganglia and several 4 synapses on this subcortical pathway. But there are also direct cortical-cortical projections. So the question is, why do you need the subcortical path at all? Why not just simply learn this way? Well, there's another feature which makes that cortical-cortical theory plausible, is that there is a massive dopamine projection into frontal cortex. So if the first trial in a task is a correct response, cortical dopamine levels will be elevated, just like basal ganglia dopamine levels. But there's one big difference between cortical dopamine and basal ganglia dopamine, and that's reuptake. Turns out there is no dopamine reuptake agent in cortex. There is in basal ganglia. So in basal ganglia, the dopamine goes into the synapse. It gets cleared out very, very quickly. So quickly, you can't even measure dopamine release in the striatum using microdialysis, which is a standard technique for looking at dopamine release. But in cortex, because there's no reuptake, it just has to diffuse out. There's enzymes that eventually break the molecule down. But if you give a hungry rat one pellet of food, you can actually measure elevated cortical dopamine levels for 20 minutes. So the first trial, correct response, cortical dopamine is going to be elevated. That's going to strengthen maybe the synapse that was responsible for that first response, but it's going to remain elevated throughout the whole session. So if the second trial is an error, dopamine's still high, it's going to still strengthen active synapses on the second trial. And that kind of a system can't learn these arbitrary mappings. So how can we solve this problem? Well, what I've argued is that what happens, for example, if we present a member of category A, the basal ganglia system, because it has this very quick trial-by-trial up and down dopamine signal, can learn these associations. And what it's going to do, it's going to activate the correct postsynaptic target in the motor areas. Now, if you think standard heavy and learning algorithms, where we strengthen synapses, not whether they're correct or incorrect, but simply as a product of the pre- and postsynaptic activation, if I somehow could activate the correct postsynaptic target, there's going to be a greater product of pre- and postsynaptic activation at the correct synapse than at the incorrect synapse. And that will allow standard heavy and learning, two-factor heavy and learning, to train up the right projections, cortical projections. So the argument then is that the subcortical path is only there to activate the correct postsynaptic target. It's going to then strengthen correct cortical projections. And this system is eventually going to take over because it's faster, and you don't need all those synapses in the subcortical pathways. So that's the model. This came out last summer. You got to have an acronym. So this is subcortical pathways enable expertise development. And basically, it's the same old learning model, but the learning model is activating the correct postsynaptic target. So what we did is we instantiated this as a computational model, and this is the architecture on a task where there's two categories, A and B. We only have 2 units in the striatum. We have 10,000 units up here, but each of these striatal units has 10,000 spines. Lateral inhibition here and here. And I won't go through these equations, but we essentially just model the activation in each one of these units using differential equations. So you can think of the left-hand side as something like the firing rate of the cell. Positive terms increase the firing rate, negative terms decrease the firing rate. So mainly what we're modeling is the input and output relation shown here, whether each input and output is inhibitory or excitatory. And all this neuroanatomy is known, so there's nothing speculative about that. What's speculative is the function that we're ascribing to it. And we also modeled decay, so that if you turn off all the inputs, activation decays to baseline. We modeled saturation so that firing can't go above 1, can't go below 0. But that's really all these equations are doing, and we have equations for all the areas. Now, the other thing that we have to model that's important is learning. And a critical feature of this model is there's learning at 2 different kinds of synapses. There's learning at cortical striatal synapses, and we're arguing that's 3-factor learning. This is what they call it in the neuroscience literature. But basically, the 3 factors you have to strengthen the synapse: strong pre- and postsynaptic activation, and dopamine above baseline is the 3rd factor. And then we have learning at cortical synapses, but this is 2-factor learning. So now any active synapses get strengthened, regardless of whether there was a correct or an incorrect response. Okay? And the way we modeled them, this is the cortical standard heavy and learning. Well, it's not quite standard. It's almost standard. This is the strength of a synapse between 2 cells. The one thing that we added, because there's lots of neuroscience evidence for it above and beyond typical heavy and learning, is that we added a postsynaptic NMDA receptor threshold, and that's because there's a lot of neuroscience data showing that you only strengthen the synapses if NMDA receptor activation occurs postsynaptically. And we weaken the synapse if there's weak postsynaptic activation, that is below the NMDA threshold. The three-factor learning, we strengthen the synapse. Now there's three factors required. We need strong pre- and post-synaptic activation, again, above NMDA receptor threshold. But now dopamine has to be above baseline. So D of N is the actual amount of dopamine released on each trial. And we weaken the synapse. If you have strong activation, but the dopamine's below baseline, that's going to be an error term, an error trial. And we also weaken the synapse if you have weak postsynaptic activation, regardless of whether the response was correct or incorrect. Now, another critical thing, the last critical piece here, the model requires that we specify exactly how much dopamine is released on each trial. And there's been a lot of work on this in the literature, and many of you probably know that there's a lot of work out of Schultz's lab and others showing that the amount of dopamine release increases with the difference between the obtained and predicted reward. So the first paycheck you ever get, you open the paycheck and you're really, you're not sure what's going to be there, and you see this big number and you're really excited, it makes you really happy, but after the 100th one, you don't even open the paycheck anymore, right? You don't open the envelope. The idea is that expected rewards don't cause dopamine changes, only unexpected rewards. And that's reflected in our model. So this is actual dopamine cell data that came from Baron Glimscher. And this is the model that we use to model dopamine release. Incidentally, this is just the amount of dopamine released by the model on each trial of a task when the model is learning these information integration categories. And what you can see, the dopamine baseline is 0.2 in this example. And you can see initially there's wild fluctuations up and down in dopamine levels because initially the model doesn't know whether it's going to be correct or incorrect. And correct responses, push dopamine way up. Errors push dopamine way down. But eventually, things become more and more predictable. Out here, the model's correct almost all the time, and dopamine's just staying at baseline because it knows it's going to be correct. There's a few cases here where it's surprised. But from the machine learning perspective, this is actually simulating the machine learning phenomenon of cooling, where many people add cooling, simulated annealing and cooling to their models. Dopamine is acting as a natural annealing and cooling agent. So how's the model going to learn these categories? If you actually, these are actual simulations or solutions of those differential equations. So this is about 50 trials into learning. And we present a stimulus from category A. It's driving, I've only shown the A unit here, the A pathway here, but it's driving up activation in the A striatal unit. You can see activation goes up. Now, the striatum is inhibiting the globus pallidus, so when it goes up, it shuts down the globus pallidus. But globus pallidus, it turns out, has very high spontaneous firing rates. So the globus pallidus is firing very, very quickly until the striatum turns on, and then it gets pushed down. Globus pallidus is inhibiting the thalamus, and so when it gets suppressed, the thalamus is released from this tonic inhibition, and it's allowed to respond to other excitatory inputs, which I don't have shown in this figure. And then the thalamus excites the motor areas, and it causes the motor unit to start firing, and then the model presses the A button. Now, this is the same exact simulation, but this is about 2,000 trials later. And the main difference now is that you can see that this motor unit is being driven to above threshold before the thalamus is even released from its tonic inhibition. And that's because this cortical synapse has been strengthened enough that it's driving the motor unit. And I'm going to skip some of this stuff. This is just showing that the model can't learn without striatal noise, which is a really interesting dynamical systems thing, but I'm going to, in the interest of time, skip that. And this is showing the benefits of having the NMDA threshold in the model. I'll skip that too. Okay, so this is the monkey data I showed you before, and we modeled that data. This is actual learning, psychometric function for the animal. This is the 10 speeds, and this is the animal's accuracy. This is the model. The model does a good fit to the human or to the monkey data. This is the model's striatal units. This is the data I showed you before from the monkey. Here's the model. So the model is really doing the same thing. It's developing these category-specific responses. This is the model's premotor cortex, and Romo and Merchant also recorded from premotor cortex in their experiment. These are the histograms of cells that show this is low-speed category-sensitive cells, high-speed category-sensitive cells, and here's the histograms from the model. But I want to show you this is some other data I haven't talked about, which is really interesting. So this is a rat study, and a rat's in a box. When it here's a tone, it should go press a bar, and it learns eventually that if it presses the bar within 10 seconds of the tone, it's going to get a food reward. It gets 70 trials a day and 18 days of training. It doesn't take the animals 18 days to learn this. They learn this in a day or two, but they kept overtraining. And Corelli, Wolski, and West recorded from the striatum of the rat during this whole process. And here are striatal responses from the rat. And here's day 4. This is after, now the animal has reliably learned this behavior, and you can see that the striatal unit is firing a burst right before the lever press. And there's other data showing, in fact, that this is a causal relationship. If you lesion this area, the animals won't learn these associations. But they kept training and recording, and you can see by days 5 and 6, the striatum is still firing a burst, but it's now after the lever press. So clearly, it can't be controlling the lever press anymore. And eventually, the striatal response just completely disappears. The animal's still very reliably and quickly lever pressing down here. So here's the model. Same exact experiment. The model does the same thing because what's happening by days 5 and 6 and these later days is the behavior is becoming controlled by the cortical projections, and the striatal unit is still firing, but it's after the motor unit has already been driven above threshold by the cortical input from, in this case, auditory cortex. And eventually it quits, and that's because the reward's completely predictable, so there's no more dopamine. It stays at baseline, and we have a slow decay in the strengths of the synapses in that case. Last study here, we did some other ones, but I'm going to skip some of them. This is a study by Rob Nosofsky and Tom Palmieri. Here's your color naming, except it's color categories. These are arbitrary color categories. These are all well, they're supposed to be iso-hue. They probably aren't in this slide, but they're supposed to be iso-hue, varying in saturation and brightness. Arbitrary category boundary. 3 human subjects, got 1,800 trials. The primary focus of this paper was in the response times, not in the accuracy. All the subjects achieved perfect accuracy. So I won't even show you the human accuracy data. But here's the model. The model learns the categories fine. And all we do is we input the actual numerical Munsell values into the program. Again, the focus in this paper was the response time. And one of the phenomena in human automaticity work is called the power law speedup. And that is if you practice any behavior, and this has even been done on cigar rolling. There's a famous cigar rolling paper showing that the more you practice, the faster you get. But not only that, the improvement in speed is very well accounted for by a power law. This is one of Nosofsky and Palmieri's human participants. Here's this RTs predicted by the model. And the solid line is the fit of the power law speed up. So it's behaving in the same way that humans do. These are the RT density functions from the model. And the reason I'm showing these, there's a couple of reasons. First of all, notice that, of course, not only is the mean RT decreasing, but the RT variance is also decreasing. And there's a lot of work showing that that's exactly what happens with human learning, is that the responses not only become quicker, they become less variable. And the other thing I want to show is that the model does not produce bimodal distributions, even though there's these 2 pathways, because it's not an either-or. It's because the motor unit needs multiple spikes in order to exceed threshold. And what happens is a gradual transition. And so there's a period of time when maybe the cortical is giving a couple of spikes, and then the subcortical comes in and gives 1, and all 3, both pathways together, driving the thing above threshold. And that's why you get unimodal distributions here. All right, so I'm going to just close up real quickly here. So there's a lot of interesting future thing. One thing we're doing now is Fmri. So we've got subjects we're training for 20 sessions with the information integration categories. So thousands and thousands of trials of practice. And then we just got a scanner at Santa Barbara. So days 2, 4, 10, and 20, they go down and they repeat this learning process in the scanner. The model clearly makes very strong predictions about how not only which brain areas ought to be active, but how those activations ought to change as a function of practice. We could model different neural site groups and effects of different drugs, which I'll skip here. But another thing we need to extend the model to is what happens in rule-based tasks. How do those become automatic? And there's actually some data that came out a year or so ago in Journal of Cognitive Neuroscience where they trained monkeys to apply rules for a whole year. And they recorded from prefrontal cortex and premotor cortex, and they found cells in prefrontal that would fire to the rule anytime the rule was applied, regardless of what the response was, and then other, of course, motor units that would fire before the motor response. And initially, just as you'd expect, the prefrontal rule units would fire right before the premotor cells would fire. But after a year of training, prefrontal rule cells are still firing, but they're now firing after the motor cells. So again, there's a suggestion that in rule-based tasks, a similar phenomena might be occurring. Okay, so just to conclude, I've argued that there's 2 category learning systems. Actually, I think there's probably more than that, but I just focused on 2 today. One's explicit logical reasoning system, another procedural learning system. And that the primary function, we think, of these systems is really just to train up this much faster cortical kind of representation. So thank you.
We have 5 minutes or so for questions.
Yeah.
So I think there's a phenomenon in animal operant conditioning in which allows animals to tolerate the delay between when they make the response and when they actually get reinforced, and it's a marking procedure. So I'm trying to sort that out. So if an animal makes a response and there's a sort of a neutral signal that somehow marks the response as being special, then in fact they can tolerate a delay in terms of the reinforcer that they get. It sounds as though the process that you're talking about in these sort of automatic learning systems won't tolerate delays.
Well, actually, I would think that it would work very similar way. Now, if you notice, when I talked about that study, we had the mask. Turns out you have to have a mask. If there's no mask, people can tolerate delays also. And of course, there's a lot of evidence that imagery activates the same sensory projections. And so we think it's probably because they can maintain, using imagery, the same, keep the same synapses active throughout the delay period. So you have to have the mask in there to disrupt that kind of a process to get the interference effects. And so I would suspect that that's probably what's going on. Yeah, Jack?
I'm thinking about what are the implications of this for extinction, for unlearning the thing that's now been cortically.
Well, yeah, we actually have just very recent work, which we haven't. We're just writing up now. I have to go back through this whole thing, but it turns out it's a very interesting question because one of the, you learn these behaviors, right? But let's suppose that we don't see too many of these discs in the real world, but maybe if you work in your lab or another vision lab, people see lots of these Gabors. But that doesn't extinguish the learning that they did in our lab, right? They can learn in our lab, go next door, they see the same stimulus, they don't make a response, why don't they extinguish? Well, it turns out there are interneurons. Okay, so here I have an interneuron. This is a GABAergic interneuron. So in primate basal ganglia, 96% of the cells are medium spiny cells, these guys. 2% are GABAergic interneurons, which we have here. But then there's another 2%, which are cholinergic interneurons Those are called, if you know basal ganglia work, those are called TANs, T-A-N-S, which stands for tonically active neurons. It turns out that the TANs tonically inhibit the medium spiny cells. So when the TANs are firing, and they actually fire in the absence of any input, they just have these leaky ion channels which are constantly firing. So the TANs are inhibiting the medium spiny cells, which means that if there's a activation sensory cortex, when the TANs are firing, there won't be any postsynaptic activation. In the absence of postsynaptic activation, there's no more decay. There's no LTD at the synapse. Well, there's other work showing, in fact, that the TANs learn to pause to motivationally salient cues. But they also have incredibly broad tuning curves, if you can call them that, because they'll pause not just to visual cues, but to auditory cues, to any cues. So we've actually augmented this model by adding TANs, and TANs get their input from perivesicular nucleus in the thalamus. So that what happens, you bring the animal or the subject into the lab, they start getting rewards. The TANs learn, okay, there's rewards here, so they learn to pause. They pause, that allows everything that I talked about today to go on. Now I walk next door and I see Gabor's incidentally, but there's no reward. The TANs quickly learn, okay, there's no reward anymore. They quit pausing. And when they quit pausing, there's no more postsynaptic activation. So the absence of reward doesn't cause them to lose these memories. And one of the nice things about that model is it accounts for the fact that reacquisition following extinction is faster than initial acquisition. So, but that's another talk.
Okay, if there are no other questions, I thank our speakers.